Hans Krebs, the guy who discovered this, occurs in the matrix of the mitochondria, begins with the addition of 2 carbon acetyl groups to form that acetyl-CoA, which is a 4-carbon molecule, forming a 6-carbon molecule, citric acid. The NADH and the FADH2 capture these energy-rich electrons, and ATP is formed by what's called substrate-level phosphorylation. This turns twice for 1 glucose molecule, once for each pyruvate. This produces 4 CO2s, 2 ATPs, 6 NADHs, and 2 FADH2s per glucose molecule. Alright, again, there's your citric acid cycle, or the Krebs cycle. So now we've got another step. This is our 3rd step. And we have a net gain of another 2 ATPs. The electron transport chain. This happens in the cristae mitochondria in eukaryotes. Okay, for aerobic prokaryotes, it happens in the plasma membrane. Through a series of carrier molecules, energy-rich electrons pass successively from one to another. Through a complex array of proteins and cytochromes. Cytochromes are proteins with heme groups and essential iron atoms. The electron transport chain receives those NADH and FADH2s and produces ATP by oxidative phosphorylation. The oxygen serves as the final electron acceptor. That's why we need oxygen. Oxygen combines with the hydrogen ions from the water. The fate of the hydrogens from the NADH deliver enough energy to make 3 ATPs. Those form FADH2. They have only enough ATPs or spent hydrogens to combine with oxygen. The coenzymes are recycled to increase efficiency, but once the NADH delivers hydrogens, it returns to NAD plus to pick up more hydrogens. However, the hydrogens must combine with oxygen to make water. If oxygen runs out, okay, or if oxygen is not present, the NADH cannot release those hydrogen ions and no longer cycles back NADH, so the process will stop. Right, so here's your electron transport chain. Another animation that I will post. This one I won't. So the electron transport chain pumps hydrogen ions from the matrix to the intermembrane space of the mitochondria. This is chemoosmosis. The hydrogen ions become more concentrated in the intermembrane space, creating an electrochemical gradient. Remember, we talked about these gradients. This is what causes things to flow when there's high and low, because we want to reach equilibrium, so there is a mechanism for the movement of molecules. And now we have this electrochemical gradient because we have all of these positively charged hydrogen ions on one side of the membrane. The ATP synthesis allows the hydrogen ions to flow down its gradient from high to low. The flow of hydrogen drives the synthesis of ATP from ADP and inorganic phosphate by the ATP synthesis. This process is called chemoosmosis. The ATP production is linked to the establishment of the hydrogen ions gradient. ATP moves out of the mitochondria and is used for cellular work. So the ATP is being produced through this chemoosmosis because there's a electrochemical gradient that the hydrogen ions are passing through. And we kind of likened it to the turbines of a hydroelectric dam. You've got your hydrogens flowing through and then generate the ATP out of ADPs. It's a kind of a poor analogy, but it gives you just an idea of high concentration driving energetic reaction. Once the ATP moves out of the mitochondria and is used for cellular work, it can be broken down to ADP. All right, that's when it's spent. and an organic phosphate, then these molecules return to the mitochondria for more ATP production. It's just a diagram of that. We've looked at this proton pump animation several times before. Again, I'll put a link on Blackboard so you can take a look at it. So the overall energy yield of the entire process, you've got your glucose molecule going in, glycolysis gives you 2 ATPs, the citric acid cycle gives you another 2 ATPs, and the electron transport chain gives you between 32 and 34 ATPs. That means that a glucose molecule has 686 kilocalories, or kcals in it. The energy yield of 36 ATPs is only 263 kcals. Alright, so now where did we lose here? This means that glucose is only 39% efficient. The rest of the energy from glucose is lost as heat. Okay, this is important and this is interesting. Glucose, if you break it down entirely, can produce 686 kcals. But the energy yield from 36 ATPs is only 263 kcals. You only get 39% of that energy. So when you're doing a physical activity, when you're burning energy, right? You're metabolizing, your cells are undergoing cell respiration. You get hot. Like if I had you stand up now and start doing jumping jacks, you're going to start building up body heat. That body heat comes from the expenditure of those glucose molecules and breaking them down. And again, you're losing a significant amount of heat. 61% of the energy available in a glucose molecule breaks down as heat. Right, so there's again a mathematical equation for the breakdown of a complete glucose molecule. So the metabolic pool, or catabolism, catabolic is breakdown. Anabolic is build. Think like the bodybuilder is anabolic. Anabolic steroids build muscle. Catabolism is the breakdown. So foods are sources of energy-rich molecules. We've got carbohydrates, we've talked about macromolecules, we've talked about fats, we've talked about proteins. So the degradative reactions, or the breakdown, catabolism of these molecules tend to release energy. Synthetic reactions, we said anabolism, build molecules. Anabolism builds, catabolism tears down. They tend to consume energy. Anabolic reactions tend to consume energy, takes energy to build, and breaking down releases energy. So the metabolic pool concept, when you eat food, it's typically not just a single macromolecule, right? If you ate this pizza, you've got various macromolecules that you're consuming, proteins, carbohydrates, and fats, and they're all broken down in that metabolic pool differently, but they all ultimately are feeding into glycolysis, because that's the only way that the full amount of the energy is extracted. So proteins are made of amino acids, okay, but they're also glucose molecules attached. All right, so the amino acids are stripped off, and then they find their way into glycolysis. Then you also have your carbohydrates, which are essentially starches that have glucoses. They're made up of glucose molecules. They feed right into it. Okay, and then fats. You've got your glycerols and fatty acids. Again, they're stripping off one part, and then the glucoses end up in the breakdown, the cell respiration process. So the metabolic pool and catabolism, the glucose is broken down in cellular respiration. Fats break down into glycerol and 3 fatty acids. Amino acids break down into carbon chains and amino groups. The amino acids sometimes are removed in the liver. This results in the ridding of NH3 or ammonia, which is poisonous. Right? It's quickly converted to urea. It goes out of our body in our urine. Different R groups from amino acids are processed differently. The fragments enter the respiratory pathways at many different points. All metabolic compounds are part of the metabolic pool. Intermediates from respiratory pathways can be used for anabolism, building. Anabolism, we said, synthetic or synthesis reactions. Carbohydrates start with acetyl-CoA, basically a reverse glycolysis but different pathway. So we can make carbohydrates too through a reverse glycolysis system. Fats, G3Ps, are converted to glycerol. Acetyl groups are connected in pairs to form fatty acids. Anabolism, or the building of proteins. Okay, this is a kind of a lead into a different lecture, but proteins are made of a combination of 20 different amino acids. All right, remember that. 20 different amino acids make up proteins. That's it. Whether you're a human, whether you're an oak tree, we have 20 different amino acids. Some of these amino acids, 11, this is important, can be synthesized by adult humans. So adult humans can only synthesize 11 amino acids. However, other amino acids cannot be synthesized by humans. Those are called essential amino acids, and there are 9 of them. Okay? There are 9 essential amino acids that cannot be synthesized by humans. So we have to have those in our diet. All right? The lysines, is one of them. I think we've talked about that in another lecture. They're limiting amino acids sometimes in the diet, but these are the essential amino acids. All right? You have to have them present in your diet because your body just can't make them. Photosynthesis and cell respiration. We started this lecture looking at how they balance each other out. We have the sun producing energy that's captured through photosynthesis and stored in the glucose molecule. That glucose molecule then goes into producing ATP for all the organisms on the planet. So for all the cellular work, this has to happen. So for me giving this lecture, for you sitting there listening to this lecture, all of the work that's happening is a part of the photosynthesis and cell respiration reaction. And it's such a harmonious equation because, like I said, the byproducts of one feed the other. And essentially sunlight is passing from one organism to another to drive that work. So thank you for listening. I'm sure this was a little bit challenging at times. If you have questions, please put them in an email. I'll respond to them as best I can. I will be posting those videos, the McGraw-Hill videos, to YouTube. You'll definitely want to start using the LearnSmarts. The LearnSmarts, I've actually attached a higher point value now to those LearnSmart activities to really help drive home some of the lecture points. So again, thank you, and we'll keep talking.